Hey everybody, thanks so much for tuning in. We are going to today be talking about the viruses, which is one half of chapter 13. If we take on the entirety of chapter 13 in one YouTube video, I'm afraid it's going to get a little bit too big and cause you some problems while you're streaming. So stay tuned for the remaining information from chapter 13, the prions. Those will come at you in a different YouTube video link very soon. So today, viruses. So even though viruses weren't discovered until much later, the term virus can be found in use as early as 1392, where it was used in England to refer to poison and other noxious substances. Likewise, the use of the word virulent, from the Latin virulentus, or poisonous, dates to about 1400. That said, the earliest mention of virus, meaning an agent that causes infectious disease, doesn't happen until about 1728. But by the mid to late 1800s, the scientific world had learned so much about microbial disease. You might remember my mentioning of the work of Robert Koch and his germ theory of disease way back in chapter 1, which was the first use of the scientific method to attribute a disease process to a particular microbial agent, with very scientific and rigid steps to connect those two being used. And it turns out that his work was pivotal not only in the understanding of disease processes, but also in the understanding of viruses, because there were a number of diseases that seemed to be infectious in nature for which no causative or etiologic agent could be identified. Now, particularly after the work of Charles Chamberlain, who was a French biologist, invented a filter with pores too small for bacteria to pass through, this became kind of a thing that was interesting for people to study. Now, Chamberlain had intended this filter to be used to sterilize solutions by removing bacteria, so physical removal of microorganisms that we talked about in chapter 7. But the unintended discovery was that there were filterable infectious agents within that liquid which could transmit disease even though they could not be cultured according to the postulates of Koch's germ theory of disease. One such agent was studied by none other than Louis Pasteur, and that was rabies. And Pasteur studied rabies and was kind of perplexed because he was unable to find the causative agent, and he speculated that it was caused by a pathogen too small to be detected using a microscope. And as we'll learn in the diversity portion of this unit, he was right. So as we wrap up our discussion of sort of the history of viruses, note that right before the turn of the 20th century, a Russian biologist named Dmitry Ivanovsky used the Chamberlain filter to study something called tobacco mosaic disease, which was a crop disease of the tobacco plant and lots of other plants, which is shown in this picture here. You can see the leaf that's sort of at 3 o'clock has this brown and yellow mottled area on it. And though it's called tobacco mosaic disease, it's found in the cash crop tobacco, but it's also found in food sources such as tomatoes and cucumbers and other things. So it was this scourge of the agricultural world. And so he started studying it, and what his work showed was that ground up leaf extracts from infected plants remained infectious after he used this Chamberlain filter, which should have removed all infectious agents. And in 1898, a Dutch microbiologist by the name of Martinus Beijnerink picked up where Ivanovsky had left off and coined the infectious agent within that liquid, contagium vivum fluidum, which is Latin for soluble living germ. So basically aqueous living germ, so infectious water. And he called the thing that was in that infectious water a virus. So there we really see a connection between virus and disease, much more clearly connected than when it was first used in the early 1700s. Now, Beijnerich thought that viruses were a liquid component, But it didn't take long before technology caught up to the virus, and it was determined that no, in fact, these were not liquid. As Wendell Stanley discovered in 1935 through electron microscopy, the causative agent of tobacco mosaic disease was in fact a particulate agent known as tobacco mosaic virus today. So not only did Stanley take an electron micrograph of the tobacco mosaic virus and determine that it was particulate in nature, he also determined that this virus was made up almost entirely of protein, which made it very different from any other thing that anyone had discovered. So he takes an electron micrograph in 1941. The tobacco mosaic virus was photographed using X-ray diffraction, and in 1955, Rosalind Franklin, who might sound familiar to you because she took a pretty famous photo of DNA, discovered the structure of the tobacco mosaic virus in 1955 from looking at the picture on the bottom left-hand side of your screen. And you might just take a moment to be wowed by the fact that this upper right-hand picture was an electron micrograph of the tobacco mosaic virus. This lower left-hand picture is an X-ray crystallograph of the tobacco mosaic virus. And somehow through all of that, scientists in the late 20th century were able to determine that the structure of the tobacco mosaic virus was what we see here at the bottom left-hand side of your screen. So pretty impressive, I would say, to come from these two pictures and then come up with this diagram where you have this not only protein, but notice protein surrounding a nucleic acid, and that protein arranged in highly specific fashion around there, which we refer to as a helix. So by the mid-1950s, we know that viruses are infectious, we know they're filterable, we know they need a host, and it seems like all of them are primarily made of protein. And in about 1950, this knowledge coalesces into a frenzy of study into viruses, which leads to the golden age of virology. So in 1906, people start growing viruses, trying to figure out how to grow them, right? Can't grow them on a petri dish, and they figure out you can grow them inside of a cell. So they're first grown in lymph fluid in 1913. The smallpox virus is grown in guinea pig corneal tissue in 1928. The causative agent of smallpox, vaccinia, is grown in minced hen's kidneys. In 1931, influenza and other viruses are grown in fertilized chicken eggs. And in the 1950s, enough poliovirus is grown in tissue culture to produce a vaccine. So the picture that you see here is a colored scanning electron micrograph of the poliovirus, which was grown in 1950 and really changed the face of infectious disease because the poliovirus had just been moving throughout the world, causing all sorts of not only deaths, but permanent disfiguring of individuals because of the damage it did to nerves in the people that survived the infection. So that was great. And then in 1949, a group of researchers led by John Enders, who's very, very famous amongst virologists. I know his name probably doesn't sound familiar to you at all, but one of his claims to fame was that he was able to grow poliovirus in human embryo cells, and that was the first virus to be grown without using solid animal tissue or eggs. And his work enabled Jonas Salk to make an effective polio vaccine. So at this point, we have two vaccines, Salk and Sabin, and we won't discuss what that means right now. We'll wait until our vaccine unit in chapter 18. For now, we will go back to our viruses, right, and see this golden age beginning in 1950, leading until the year 2000, where almost all of the known species of viruses were discovered. So of all of the almost 2,000 viruses that we know today, most of them were found during that 50-year period. Now, kind of an interesting side note to this period of discovery happened way before the golden age of virology, and that was some work done in the 19-teens by a French-Canadian microbiologist named Félix Deriel, who studied bacteriophage, right? So we're going back to that bacteriophage, that complex virus that does not infect humans but infects bacteria. And he studied these guys a lot, and he learned not only who they were, but he actually learned how to culture them within their bacterial hosts. And not only that, was able to do serial dilution and plating of these bacteriophage with their bacterial hosts in order to calculate the number of viruses in a given solution, which should sound familiar to you because you yourselves have studied serial dilution and plating, and you actually have a lab where you serially dilute and plate bacteriophage along with their bacterial host. And when he did this work in 1917, people were really excited, and they started thinking about all the possibilities that these bacteriophages might bring to the table, right? Because these bacteriophages were able to infect bacteria, people thought, you know, these could be the miracle treatment to wipe out bacterial disease like typhoid and cholera, things that were just wreaking havoc all over the globe. But in the 1920s, a miracle called penicillin was discovered, and everyone forgot about phage therapy until the 21st century, when antibiotic resistance, including resistance to penicillin, has become such a problem that we're searching out alternate therapies to cure bacterial diseases. So the tobacco mosaic virus is what we call a geometric virus. As you can see here at the bottom on the left-hand side, it's a helical virus. In addition to that helical virus, we have two other kinds of geometric viruses, the icosahedral and the polyhedral viruses. Each of them is considered a simple virus because it is a piece of genetic material surrounded by a protein coat, which we call a capsid. And that protein capsid tends to be very uniform in nature, made up of individual subunits called capsomeres. And it turns out that those capsomeres are actually what the virus uses to bind to and attach to host cells. So these are very important structures that allow these obligate intracellular parasites to actually get inside of their host cell. So as we said, these are obligate intracellular parasites, and through our looking into the golden age of virology, we know that they have to be grown within a host cell, which typically includes an animal cell, a fertilized egg or cell culture. And we have to be really particular about the kind of cell types that we grow these viruses in, because those protein capsomeres are very specific about the kinds of cells that they can bind to, because they're binding to a particular receptor on the surface of the cell. And this is something that we're going to see in real detail when we study HIV in chapter 19, but suffice it to say for right now, every cell in the body expresses different genes, which means they have different proteins on the surface of the cells, and the viruses exploit those receptors on the surface of the cells in order to gain entry into the host cell. So they're typically very specific about the cell type that they infect, although they may be pretty loosey-goosey about the host range that they will infect, which means though they may only infect respiratory epithelium, if they have a broad host range, they may not really care what kind of respiratory epithelium they infect. So you might have something like coronavirus or influenza, where it binds respiratory epithelium, but it binds respiratory epithelium of humans and animals, right, with influenza, pigs and chickens, and with coronavirus, we have bats and things like that. So very specific about the cell type, not so specific about the host, and so specific, in fact, about the cell type that respiratory epithelium would be a target, but something like digestive tract epithelium would not be. So they can be very, very specific about the cell type they will infect and not so particular about the kind of host that they will infect. And just as a point of fact, you'll oftentimes hear people talk about virions. Virions are the infectious virus, which may seem like a matter of semantics, and in lots of cases it is. However, in some cases, a fully put together virus has to be activated in some way in order to become infectious, and so we would then make a distinction between the fully manufactured virus and the turned on virus, virion. And again, HIV is an example of that. There's a protein on the surface of HIV that has to be activated before HIV virus can become HIV virion and become infectious to the next cell. So that's sort of the nuts and bolts of viruses, simple viruses, kind of funny. We also have something called a complex virus, and complex viruses, though they are complex, infect the most simple organisms, and that is the bacteria, right? So the complex virus is the bacteriophage that we've been talking about, the bacteria eater, that filterable agent that was added to bacterial cultures in the 19-teens and seemed to make those cultures go away as if they had been eaten. And we call that a complex virus because it has multiple viral morphologies. Notice it has a capsid head that has that icosahedral or polyhedral shape, and it has a sheath that has a helical shape to it. So you have something here that's comprised of multiple morphologies, and as I mentioned, it infects bacteria, which is shown in this transmission electron micrograph where you have 3 bacteriophage colored green on the surface of a bacterial cell, which is yellow, and you can just make out the green DNA from the bacteriophage being injected into the bacteria in order to start viral infection of that bacterial cell. So we have simple viruses, we have complex viruses. We also have something that we talked about in chapter 7, and that is enveloped viruses. So enveloped viruses have an additional layer to that nucleic acid surrounded by a protein capsid, and that additional layer is a lipid bilayer envelope that they acquire by stealing it from their host cell. And when they have this lipid bilayer envelope, because proteins on the virus are always what interact with the host cell, they also have protein spikes. So protein is always going to be what binds to the host cell, either the capsomere protein of the capsid in a non-enveloped virus or the protein spike of an enveloped virus. Always going to be a protein, always going to be a protein binding to a very particular receptor on the cell. And with envelope viruses, the good news is, as a lipid bilayer envelope, all you need to do is dissolve that envelope with soap, and it renders the virus ineffective because as you dissolve the envelope, the protein spikes fall away, and the virus cannot use capsomere proteins on the capsid in order to bind its host. So it's rendered ineffective, which is why the enveloped virus is one of the least resistant of all of the microbes to control, which we learned about in chapter 7. So viruses are not alive. Notice, no cells, no ribosomes, none of the things that are used in taxonomic classification or identification that we learned about in chapter 10. So the obvious question then is, how do you classify viruses and how do you identify viruses? How do you do those things that we talked about in chapter 10 that are so important to categorizing and understanding an unknown microorganism? So we need to talk about that for a moment because it's a totally different scheme for classification and in most cases identification of viruses, most particularly the classification of viruses. And there are actually 2 methods of classifying viruses. The sort of authority today is the International Committee on the Taxonomy of Viruses, or the ICTV, which authorizes and organizes the taxonomic classification of and the nomenclature of viruses. And they have this universal scheme which relies on phenotypic characteristics, including morphology, nucleic acid type, right? Is it an RNA virus? Is it a DNA virus? Mode of replication, how do they replicate themselves inside the cell? And then finally, what is their host organism and what kind of disease do they cause? So they're sort of attempting to make order out of chaos, and it's really difficult when it comes to viruses. But what you're seeing here is one of the publications by the ICTV from 2005, I believe, where they tried to separate all of those viruses out based on these characteristics. A more simplistic approach to viral classification, I think, can be found in something called Baltimore classification, which was put together by, I believe it's John Baltimore in the 1970s, and his classification scheme is based on just two things, genome and mRNA synthesis. So he takes all viruses, any unknown virus, and you can put it into one of these 7 classes, right? Class 1 is a virus that has a double-stranded DNA genome, class 2, single-stranded DNA, class 3, double-stranded RNA, class 4, plus strand single-stranded RNA, which is the equivalent of messenger RNA, class 5, negative single-stranded RNA, which is the complement of messenger RNA, class 6, which are single-stranded RNA viruses which use an enzyme called reverse transcriptase, and then finally class 7, which are DNA viruses, double-stranded DNA viruses, which use a reverse transcriptase. Not that you need to know any of those particulars. What I would like you to know is they're based on genome and how they make messenger RNA from that genome, and sometimes that takes a viral enzyme. And that happens when the virus needs the cell to do something that the cell is not equipped to do, right? So if you think about what the cell is equipped to do with DNA and RNA, what the cell is equipped to do with DNA is to replicate it using a DNA polymerase and transcribe it using an RNA polymerase. And we talked a little bit in chapter 8 about how we can be more specific about those two names, right? The DNA polymerase that makes DNA from DNA is called a DNA-dependent DNA polymerase. DNA polymerase indicating to you what it's making, DNA dependent indicating to you what its template for that molecule is. And then likewise, double-stranded DNA can be used to make messenger RNA through transcription using an enzyme called an RNA polymerase, which in this case uses DNA as a template, so we call that a DNA-dependent RNA polymerase. So notice group 1, you got no problem there, right? You have a double-stranded DNA genome, a cell would know exactly what to do with that. Now, in a case like group 3 or class 3, you have a totally different situation. The virus is coming in with double-stranded RNA, and your cell would have no idea what to do with RNA other than translate it. So that virus would need to bring with it RNA-dependent RNA polymerases in order to make messenger RNA. So if you look at this table from your textbook, table 13.4, you'll notice they're all separated based on the nucleic acid. And then I've put in there for you the viral groups or classes, and notice the special features of biosynthesis for some of these indicates to you that there's a viral enzyme involved, right? So for instance, group 3, which we just mentioned, which is a double-stranded RNA virus, the viral enzyme copies a negative strand RNA to make messenger RNA. So that virus has to code for, in its genetic material, not only all of the structural proteins that it needs to make itself, but it also needs to code for that RNA-dependent RNA polymerase. So as we're looking at particular viruses in the diversity unit, there are going to be several examples where you see a viral enzyme brought by the virus in order to accomplish its own replication. So classification, either based on the International Committee on the Taxonomy of Viruses phenotypic characteristics, and the Baltimore classification based on genome and messenger RNA synthesis. Now, thankfully, identification does have some overlap with the organisms we discussed in Chapter 10. However, the primary mode of viral identification remains its cytopathic effects. What are the signs and symptoms of disease? And you might be interested to know, right? That's one of the ways that we're differentiating between rhinovirus, which causes the common cold, and coronavirus, which causes COVID-19. Initially, people are looked at for the signs and symptoms of disease. Do you have a fever? Do you have body aches? Do you have shortness of breath? Do you have involvement of the upper respiratory tract, like the nose and the eyes and things like that? Because those are going to tell you, based on signs and symptoms, whether you have rhinovirus or coronavirus. So cytopathic effects are really helpful with viral identification, and those are followed up and confirmed with serological tests. Remember, serological tests from Chapter 10 involve antigen and antibody interaction and may include Western blots and immunoassays such as ELISAs, which we're going to talk a lot about in Chapter 18. Right now, all we care about is cytopathic effects, serological tests, and then much like other things in Chapter 10, nucleic acid tests can be used for viral identification, including restriction fragment length polymorphisms and PCR. And what you see here on the right-hand side of your screen is a Western blot looking for viral proteins. So in lane 1, which goes vertically down from number 1, you have a positive control. These are all of the proteins found in HIV. Lane 2 is a negative control, just no proteins. And then there are 3 possible infections, patient A, patient B, patient C. And you look at those banding patterns and you determine whether or not they are indicative of HIV infection. So if you look at the table on the bottom there, if you have a P31 or a P24 and a glycoprotein 160 and glycoprotein 120, you have a positive test. So patient C would be positive for HIV, and any other banding patterns would be indeterminate, right? So those would not necessarily be negative. They would be asked to retest probably at a later date when their viral load was a little better established. So identification does have some overlap with the organisms we discussed in Chapter 10, with the exception of cytopathic effects, which are used to identify viral infections. A couple more terms that we want to talk about with viral identification before we wrap up this discussion of viruses, and that is how a viral infection plays itself out in a host. And there's a couple of different scenarios that can happen. The first, which is shown in red, is where a person contracts a viral infection. The number of viruses goes up really, really high, really, really fast, and then they are cleared by the immune system, and that's called an acute infection. And it doesn't mean that it's necessarily a very dangerous infection. It just means, as we're going to learn in subsequent chapters, that you have a rapid onset of symptoms and a rapid clearing of disease. So acute infection means you got sick quickly, you got over it quickly. Now, there are some things that you get sick quickly and you seem to get over quickly, but they actually are not gone from your body. And if those things remain at a low level in your body, as is shown by this blue line here, and then later on can crop back up and cause a secondary infection, we call those persistent infections. And a persistent could be HIV, could be a particular kind of measles infection where you thought you got over the measles, but you ended up with something called subacute sclerosing panencephalitis. So you have an infection, you think it's gone, and that infection just sort of creeps up over time, and those tend to be fatal, as opposed to latent infections, which would follow the graph of the red acute infection, and then show no signs or symptoms and no real measurable viral load, and then would pop back up in this green curve here as a latent infection. So we have acute. If acute is not cleared completely, you can have a persistent infection, you can have a latent infection, and both persistent and latent infections are considered chronic infection, right? Something that you don't clear. So latent infection could be something like chickenpox, for example. The red curve could be chickenpox, and then the green curve could be a secondary chickenpox infection, which we would call shingles. So latent and persistent infections. All right, now, how do viruses replicate, right? Because we know how cells replicate, mitosis, we know how bacteria replicate, they use binary fission, but how do bacteriophage and other viruses replicate? And I think the easiest way to see that is to watch that happen dynamically. So let's take a look at bacteriophage replication.
Virulent phages carry out a simple life cycle called the lytic cycle. The phage attaches to the host cell during the attachment stage and then penetrates the host cell and injects its DNA during the penetration stage. After penetration, the phage causes the host DNA to break into small pieces. The phage then uses the host machinery to synthesize new copies of its DNA. This process is part of the biosynthesis stage. Biosynthesis also involves production of viral proteins. Once biosynthesis is complete, the phage components are assembled into virions during the maturation or assembly stage. In the release stage of the lytic cycle, the cell lyses, releasing the phage virions. These released phages go on to infect other cells.
And as we know from our therapeutic phage typing lab, the result of those infections is something called a plaque, where a single virus has infected a single E. coli cell and erupted out and caused the infection of neighboring cells. And so we refer to that virus that was able to cause that plaque as a plaque-forming unit. Now, for reasons that are not always understood, viruses infect bacterial cells and don't undergo this lytic infection where they undergo sort of clockwork-like structure of attachment, penetration, biosynthesis, assembly, or what's most commonly called maturation, and then release. They move into what's called a lysogenic cycle. In a lysogenic cycle, if you compare that to the lytic cycle that's shown on the left, where you have attachment, penetration, biosynthesis, maturation, and release, you instead have, at step 2, once the DNA has penetrated the cell, it incorporates itself into the bacterial chromosome, at which point it's called a prophage. And though it doesn't cause massive viral replication, it does allow for the viral DNA to be replicated every time the bacterial cell undergoes binary fission, which, as we know from chapter 6, could be as often as every 6 minutes. It's unclear why that happens. It's unclear why that can also come out of the lysogenic cycle and move back into the lytic cycle. But one thing that is very clear is that the ability of these viruses to incorporate themselves into the bacterial DNA can cause increased virulence of those bacterial species. For instance, one real well-known one is the incorporation of viral DNA into Streptococcus pyogenes, which takes a normal run-of-the-mill strep throat infection by Streptococcus pyogenes and turns it into scarlet fever, which is something we're going to be talking about more during later chapters when we talk about pathogenicity in Unit 3. So this idea of viral replication existing in these 5 sequence in this 5-sequence paradigm, attachment, penetration, biosynthesis, maturation, and release, is found in not only bacteriophage, but also in other viruses. And I just want to take a quick look at animal virus replication, not necessarily because you need to know the details of it, but because, again, it's nice to see those processes happen in dynamic, moving fashion rather than seeing a picture of them.
Animal viruses vary based on the type of nucleic acid they possess, as well as whether they are naked or enveloped. Each variance results in different types of life cycles. A naked and an enveloped virus will attach to and penetrate a host differently. Animal viruses without an envelope are referred to as naked viruses. They bind to the surface of the host cell and inject their DNA into the host cell in a manner similar to bacteriophages. Some envelope viruses infect their host by binding to receptors on the host cell. The envelope of the virus then merges with the host membrane and the capsid enters the cell. After entry, the capsid opens and releases the viral genetic material into the cytoplasm of the cell. Upon attachment, some envelope viruses infect their host by inducing phagocytosis, in which the host cell envelops the viral envelope and absorbs the virus into its cytoplasm. When the virus has entered the cell, the outer and inner part of the envelope merge together, and the capsid is released into the cytoplasm. At this point, the capsid opens up and releases the viral genetic material into the host cell cytoplasm. The type of nucleic acid in an animal virus will determine how the viral nucleic acid and proteins are synthesized. When the single-stranded DNA genome of a parvovirus enters a host cell and invades its nucleus, a complementary strand is produced for the single-stranded viral DNA. This DNA is then replicated normally. Messenger RNA is transcribed and transported into the cytoplasm, where viral capsomere proteins are produced. At some point after synthesis has begun, the capsomere proteins enter the nucleus of the host, and the virions containing the original single-stranded DNA are assembled. In a virus with double-stranded DNA in its genome, the DNA also migrates to the nucleus, where it's replicated by the host enzymes in the same way that the host's DNA is normally replicated. Messenger RNA is made from the viral DNA in the nucleus, and viral capsomere proteins are made in the cytoplasm from the messenger RNA. The capsomere proteins enter the nucleus, and the virions are assembled in the host nucleus. After assembly, the virions accumulate in the cytoplasm and prepare for release from the host cell. Some viruses have a single-stranded RNA genome that can act directly as messenger RNA. This strand is called the positive strand or sense strand RNA. This messenger RNA is read by host ribosomes to make viral proteins. The virus carries a unique RNA polymerase that makes a complementary negative strand, also called an antisense strand, from the original positive strand. There is no known animal equivalent of this RNA-dependent polymerase. The negative strand RNA can act as a template for more positive strand RNA. Positive strand single-stranded RNA viruses are assembled in the cytoplasm of the host cell when the RNA and the proteins have all been produced. Viruses with single-stranded negative sense RNA are in a special situation. Their RNA will not act as messenger RNA until it's been transcribed into the positive sense strand complement of the original negative antisense strand RNA. To accomplish this feat, the viruses carry their own RNA-dependent RNA polymerase, which transcribes the RNA into the positive strand. When the positive strand has been produced, it acts as the template for both protein synthesis and more negative strand RNA. When the virion is packaged, it must contain the negative strand RNA in addition to copies of the RNA-dependent RNA polymerase. When double-stranded RNA viruses infect a host cell, the positive, sent strand of the RNA acts as a messenger RNA for protein production. One of the proteins coded is an RNA polymerase that uses the negative strand to make more positive strand RNA. The positive strand also acts as a template to make more negative strand RNA. The two strands come together and are packaged along with the polymerase in the virion. Retroviruses make up a special class of positive strand RNA viruses. When a retrovirus infects a cell, its positive strand RNA is reverse transcribed into a DNA negative strand by a viral reverse transcriptase. This negative strand DNA is further transcribed to double-stranded DNA, which serves as a template for the viral RNA genome. This positive strand RNA also serves as the template for viral protein synthesis. The reverse transcriptase is packaged with the mature virion as it's essential for the successful reproduction of the virus. Just like the beginning of the life cycle, naked viruses differ from envelope viruses in how they assemble and release. Naked viruses generally leave their host cell by accumulating to such numbers that the cell lyses, spilling virions into the extracellular medium free to infect new hosts. This kills the host cell and generally causes inflammation and infection in the tissue where the host cell is found. Envelope viruses leave the host cell by merging with one of the cell's membranes, the nuclear membrane, the endoplasmic reticulum, or the cytoplasmic membrane. During synthesis, some viral glycoproteins are embedded in cellular membranes, and these glycoproteins act as a recognition system for the viral capsid. The cell membrane surrounds the capsid and forms a bud on the outer surface of the cell, which pinches off and forms a protective envelope for the virus, which is then free to infect other host cells. An advantage of the envelope is that the virus does not need to break the cell to be released, so the cell can stay alive longer while it continues to produce and bud off more viruses. The type of nucleic acid, along with the presence or absence of an envelope, will determine the type of life cycle a given animal virus will have.
When an enveloped virus attaches to its host cell, oftentimes a little something different happens, and that is that the viral envelope interacts with the cell membrane of the cell and either allows the virus to be endocytosed, or, as is the case with the herpesvirus that is shown in the sort of middle of your slide, the envelope will fuse with the plasma membrane, and then the capsid portion of the virus will go into the cell. So a little bit different on the attachment and penetration side, also different on the release side. Rather than the cell blowing up, they release by budding, and they will collect their lipid bilayer envelope either by taking some of the nuclear membrane, some sort of membrane-bound organelle membrane, or by taking a piece of the cell membrane itself. So notice on the right-hand side, you have the viral capsid, and it's moving toward the surface of the cell, and all these viral proteins are coalescing in one area of the cell membrane, and then the virus actually buds out of the cell and is surrounded by this piece of the membrane, which is now its envelope, and also has a lot of viral protein. Remember those spikes, which are going to be so important for it to bind to its host cell as it leaves this cell which it has already infected. That is where we are going to end this lecture. We will pick back up with other information as we move through Chapter 13, Part B in the prion section of the chapter.